So welcome to the faculty lecture series. Tonight we have the pleasure of listening to Juan Carlos de la Torre, molecular virologist par excellence, who ever since he came to Scripps in 1989, I believe, has been teaching me more about viruses than almost anybody else on campus. And it's a real pleasure to introduce him tonight. As I'm sure everybody knows, to really succeed in science, you have to be reasonably smart. But you have to work really hard and be savvy enough to pick really important questions to ask, and to have luck on your side as often as possible. And you have to do at least one other thing, and that's to pick really good mentors. Now, Juan Carlos has succeeded on all of those fronts. And for example, what he really did was he picked 3 fabulous mentors to help him get started in science. Juan, Esteban Domingo in Madrid, John Holland at UCSD, and Michael Oldstone here at Scripps. With Domingo and Holland, Juan Carlos studied a variety of RNA viruses that have very high mutation rates. And because of those high mutation rates, they establish in infected individuals a swarm of mutants or a quasi-species that enable the viruses to escape and evade and become resistant to the immune system and to drugs, and to become selected, and in effect guarantee that new epidemics will continuously be observed when it comes to RNA viruses in the future. A very important development. When he joined Michael Oldstone's lab, his first projects were not on arenaviruses. They were not on LCMV. He worked with another Oldstone protege named Ian Lipkin and Michael to characterize and to determine the nucleotide sequence and life cycle of Borna disease virus, and essentially established the Borna disease virus as the prototype of a new family of viruses, the Bornaviridae. That in itself is an accomplishment that very few virologists can claim to have matched in their careers. The depth and breadth and scope and implications of that discovery and that development were enormous. But of course, nobody can get out of Oldstone's lab without studying LCMV, the prototype arenavirus, the most widely studied virus that establishes persistent infection and has served as a magnificent platform to launch the careers of many different virologists, most of whom have been studying pathogenesis-related questions, immune attack of LCMV and the consequences of that immune response. And what Juan Carlos did during his postdoc with Michael was something else, something very special. In a series of landmark papers, he established that persistent non-cytolytic viruses like LCMV can interrupt luxury functions in the cells that it infects, without killing them. Luxury functions like growth hormone production, or like synaptic plasticity, or like I'll get it, cognitive functions. That was my little attempt at a joke, I'm sorry. Anyway, since joining the since joining, I'm sorry, Juan, there we go. So since joining the faculty back in 1996 and being promoted to full professor in 2009, Juan has published over 100 papers on Borna and on LCMV that have represented incredible contributions to the literature of both viruses. Perhaps most notable has been his singular development of a reverse genetic system for LCMV. That was an extraordinarily impressive accomplishment because of the structure of the LCMV genome. It's an ambisense RNA, negative strand RNA virus. It has 2 segments. And to have the technological expertise to be able to convert that into a reverse genetic system was remarkable. It was also remarkable because it enabled, for the very first time, the molecular definition of the virological outcome determinants that could be studied in vivo that determined the outcome of this persistent infection. So it was an enormous contribution. An important outgrowth of that was his development of recombinant arenaviruses with tri-segmented genomes. This enabled now the development of vaccines for arenaviruses and to study the role of individual arenavirus proteins in escape from immune pressure and in drug resistance. And arenaviruses besides LCMV, like Lassa and others. So reflecting all of these accomplishments, Juan Carlos is highly sought after as a speaker at national and international congresses and symposia. He's on the editorial board of several major virology journals. He's a member of the International Committee on the Taxonomy of Viruses in recognition of his groundbreaking Borna work. And he's been a charter member of the Virology B study section for almost continuously since 2003. Unfortunately, that means he has to leave the room when our grants are being reviewed by that study section. I'm sure he would do a very, very fair and objective review of those. So anyway, this evening, Juan Carlos is going to take us on a journey, an arenavirus journey, from the basic virological studies that he does to the therapeutic targets that he's identified. And I'm really looking forward to that. And one final word is, in the tradition of this meeting, of this lecture series, there won't be any questions afterwards, but there'll be a reception in Juan's honor in the Galleria of the Beckman building, to which everybody is invited. And you can ask him all your questions at that time. So Juan, thank you for being such a great colleague and teacher to all of us over the years, and we look forward to your talk.
Thank you, Fran, for the kind introduction. So let me set up this here for a second. So actually, as Frank indicated in the introduction, the arenaviruses, they really merit significant interest as really very powerful experimental model systems to study both acute and persistent viral infections, as well as important human pathogens. So many of you are familiar with the prototypic arenavirus, lymphocytic meningitis virus, LCMV, that it really has been and continued to be a remarkable, really, workhorse in the field of viral immunology, where actually many important concepts, including MHC restriction of T-cell responses, were first identified and characterized studying LCMV. On the other hand, several arenaviruses, mainly Lassa and Junin viruses, cause hemorrhagic fever in humans, and they really pose a significant public health in their endemic regions. And moreover, there is increasing evidence that the globally distributed LCMV is a neglected human pathogen, particularly relevant in neonatal infections and also immune-compromised individuals. I think it is worth to briefly mention the relevance in public health of Lassa, where in West Africa, in sub-Saharan West Africa, where Lassa virus is endemic, it is estimated that causes over 300,000 infections yearly, that they are associated with significant mortality and very high morbidity, and also long-term sequelae. Additional concerns about Lassa infections or any other human pathogenic arenaviruses are the fact that there are no licensed vaccines, and the therapeutic strategies currently available are limited to the use of ribavirin, a nucleoside analog that is only partially effective and associated with significant side effects. The arenaviruses are relatively simple viruses. They are envelope viruses with a bisegmented negative strand RNA virus. Each segment uses what we call a ambisense coding strategy to direct the synthesis of two different polypeptides. The S, or a small segment, encodes the virus nucleoprotein, and the precursor glycoprotein GPC that is post-translationally processed into GP1 and GP2, that they form the complex that decorates the surface of the virus and mediate receptor recognition and cell entry. The L, or large segment, encodes the virus RNA-dependent RNA polymerase, or L protein, and a small ring finger protein called Z that is a bona fide matrix protein that basically you could imagine that is the link between the ribonucleoprotein complex and the surface glycoprotein complex to really form the mature infectious particle. And also the Z protein as a bona fide matrix drive the budding process from the cells. So basically, briefly, the life cycle of this virus is relatively simple too, and is restricted to the cytoplasm of the cell. So the virus enters via receptor-mediated endocytosis. When it reaches the acidic environment of the endosome, the glycoprotein mediates a pH-dependent fusion event that releases the ribonucleoprotein into the cytoplasm, where RNA replication and transcription take place. So the glycoprotein, as I indicated, undergoes a relatively complex post-translational process to form a ternary complex between GP1, GP2, and a stable signal peptide, which is rather unique to arenaviruses. And that complex traffics to the plasma membrane, where in a process mediated by the Z protein, the matrix, interacts with the ribonucleoprotein core to form the mature infectious particles that they are released via budding from the plasma membrane. So one of a general question that we're interested is how these viruses, that they have a very low proteomic complexity, only 4 different genes, they are capable, however, to interact very differently with their infected host. So in the natural host, most of the arenaviruses, they cause a long-term persistent infection in the absence of any noticeable clinical symptoms. However, when they jump into a non-natural host, they may cause a very severe disease, including hemorrhagic fever. So we're trying to understand how the virus can regulate very differently its life cycle depending what kind of species is infecting. And the overall approach that Frank mentioned that we have been pursuing is the so-called reverse genetics. So what this means is that now we're in a position that we can recreate the different steps of the virus life cycle from cloned cDNAs. Basically, we can deliver into the cell the appropriate combination of plasmids, and eventually we can produce a recombinant virus that is going to have predetermined mutations in the genome that you want to introduce in such a way that then you can study mutation function analysis in the context of the natural infection. So one of the limitations is that many of the mutations that you are interested in may cause a very profound decrease in viral thickness, which complicates the rescue of the virus. And to overcome that, we developed also the so-called mini genome rescue system that instead of rescuing the whole virus, what you do is you use a reporter gene of interest. It could be chloramphenicol acetyltransferase, the green fluorescent protein, luciferase, whatever you want. You place that reporter gene under the cis-actin regulatory sequences of the virus, and then you provide the transactin factors via plasmids in such a way that now you can zero in into specific steps of the virus life cycle. You can focus in studying replication, gene transcription, or you can focus in study assembly without the need to having to rescue an infectious virus. So, and what I would like to do next is to briefly discuss how we have been using these approaches to dissect two different steps of the virus life cycle and how the knowledge derived from these studies have uncovered new potential ways to target human pathogenic arenaviruses. The first step is a very proximal event during the virus life cycle, is what happens once the ribonuclear protein is released into the cytoplasm. It's RNA synthesis. These viruses, negative strand RNA viruses, they bring into the cell their own polymerase attached to the ribonuclear protein complex. And in the case of the arenaviruses, we're going to go through the replication transcription cycle using the S segment as an example, but exactly the same concepts apply to the L segment. The polymerase starts to transcribe at the 3' end of the genome, produces the MP messenger RNA. In the case of the L segment, would be the L protein. And eventually, at some point, the polymerase is going to change from its replicase mode, from its transcriptase mode into a replicase mode, and will ignore this signal in the middle, a non-coding intergenic region that we have shown that is a bona fide transcription termination signal, and will copy the genome into an antigenome RNA species. And now this will be the template for both the synthesis of the GP messenger RNA as well as the synthesis of more genome RNA species. And the same applies to the L segment. So the whole thing it starts basically just with the L and MP. You don't need any other transacting factors to mediate RNA synthesis. And it starts by the recognition of by the polymerase of a specific sequence at the 3' end, the genome promoter. And in the case of the arenaviruses, one thing that is remarkable is that despite these viruses, they have been evolving for many, many years in very different species. The 3' termini are extremely conserved. Actually, the last 19 nucleotides are almost conserved among all known arenaviruses. And interestingly, the 3' has almost perfect complementarity with the 5' of the genome in such a way that there is a predicted panhandle that is formed via annealing between the 3' and the 5' termini, which is consistent with electron microscopy data that shows that inside the particle, the ribonucleoproteins are circular. So we have done an extensive mutation function analysis of the promoter, and basically the summary is that both sequence specificity, the specific nucleotides that you have there, as well as the preservation of the panhandle structure are strictly required for the function of the promoter, in such a way that if you mutate either the specificity of the sequence of the structure, you eliminate promoter activity. Interestingly, Stefan Gunter in Hamburg has done exactly the same studies with the promoter of Lassa, and the results are pretty much the same. So it is really intriguing that it appears that the arenaviruses are incapable to explore alternatives in terms of what they can use as a promoter. And this is interesting because it is quite likely that this structure can be amenable to targeting. And actually, aminoglycosides, that they are potent antimicrobials, they recognize this type of a structure. And based on that, we screened several of them. And remarkably, we found that several of the aminoglycosides arginine conjugates, we use arginine conjugates because aminoglycosides, they have very poor cell permeability. But if you generate the corresponding arginine conjugates, they enter the cell much easier. And as you can see, several of them, they have a very potent inhibitory effect on transcription, replication, and consequently production of infectious virus. So we have also taken advantage of the mini-genome system to develop assays to identify inhibitors of RNA synthesis that they may act either in the assembly of a functional ribonucleoprotein, this is this green diamond, or in the activity of the ribonucleoprotein complex per se. So you can use a reporter gene in such a way you can have a quantitative readout. And using this approach in collaboration with SIGA technology, we identify a compound called ST545 that has a very potent inhibitory activity. It's significantly better than ribavirin, that is the gold standard in the case of anti-arenaviral therapies. So the therapeutic index basically is significantly higher, is over 20 times higher than ribavirin. That is what really counts, is the ratio of how effective is inhibiting without having toxicity. So this compound actually was able to prevent the establishment of persistent infections by one of the so-called immune suppressive variants of LCMV. In the case of LCMV, there are a variety of variants that they are capable to cause immune suppression in an adult immune-competent mouse and establish long-term persistence. But as you can see, if you do treatment with this compound, the virus load decreases significantly, and then if you stop treatment at day A and you follow these mice, what you find is that later on time, those mice that they were treated with the compound, they cleared the infection. So what is happening is that the reduction in virus load is allowing the immune system of the treated mouse to really come up and control the virus and eliminate the virus, preventing the establishment of persistent infection. So one of the limitations with this kind of compound is that arenaviruses, as any other ribovirus, they have high prone error polymerase systems in such a way that mutants are being generated constantly, and therefore they can escape from almost everything that you really try to attack them with. And to assess to what extent the virus could escape from this compound, we perform just classic viral genetics. Basically, you do serial passages in the presence of increasing amounts of the compound until you select for a particular virus that is resistant to the drug. Because we initially identified the drug using the mini-genome system, we knew that the only two viral factors were either the polymerase or the nucleoprotein. So you do the genetic characterization, and eventually we found that indeed there was one single change in the nucleoprotein. It's in position 403, a change from phenylalanine to leucine that was associated with resistance to the drug. We verified this using the mini-genome, and we obtained the final proof by generating a recombinant virus via reverse genetics that had that single mutation, and that virus, as you can see, is totally resistant to the drug. So to try to understand a little bit more at the structural level how this mutation was conferring the resistance to the drug, we took advantage of the crystal structure of the LASA nucleoprotein determined by Erica's Sapphire lab recently. And as you can see, this is the cartoon of the C-terminal of the LASA MP around the area of position 403 and a electrostatic map of that area. So there is a pocket where 403 is located, and 403 is predicted to be fairly deep located into the pocket. And one can imagine that this compound can access the pocket and will be able to interact by stacking with phenylalanine, but not with leucine. So, and the interaction of the compound with phenylalanine may disrupt the local structure of the MP, affecting the whole polymerase complex. So, interestingly, there are other viruses such as Lassa and Junin that we found them to be resistant to the drug, and they have in that position tyrosine. And the possibility here is that tyrosine will create a more acidic pocket to which the compound won't be able to bind. So the interesting thing is that the other residues around these pockets are highly conserved, and therefore now this could be a real potential target for developing other drugs that they will be able to enter the pocket in the case of an acidic pocket, so to target Lassa and Junin virus. So we have been talking about what happens very early during the infection, and now we're going to be moving to the very last steps during the infection, once the virus has replicated, produced their genes, and is ready to get out. So what do you need to produce an infectious progeny? So the only thing that you need, besides the polymerase and the nucleoprotein, you need both. You need the glycoprotein and you need the Z protein. So the glycoprotein is pretty obvious, because if you don't have the glycoprotein, what you get are ball particles that they are incapable to recognize the receptor, and therefore they cannot propagate from cell to cell. And you need the C protein, because the C protein is indeed a bona fide matrix protein. So in collaboration with Kathy Borden in Montreal, we determined the NMR structure of the C protein, and it's a characteristic ring finger protein that has a very disordered C terminus. And in the C terminus, you can identify canonical late domain motif. What are late domain motifs? So they were originally identified working with retroviruses. They are called late because mutations in those motifs, they prevented the retrovirus to pinch off from the cells. And you have these beautiful EM pictures where the virus is hanging on top of the cell and cannot get released from the cell. So they come in different flavors. So the most common are the so-called PTAP or PSAP motif or PPY, and also the YXL motif. So in the case of the arenaviruses, all of them, they have something in their, like a PTAP or PPY, and some of them also, they have a YXL. Although we have done a exhaustive mutation function analysis of this motif, and these ones, they have really been verified to be critical for budding, whereas so far the evidence for the YXL, that they are within the ring motif, to be critical for budding, still the evidence is not unequivocal. So this late domain motif, what they do is they basically, they hijack the so-called multivesicular body pathway of the cell. This is a complex pathway that contains several escort complexes, and each escort complex has a variety of proteins. And what very different viruses, you're talking about a retrovirus, or Ebola virus, a filovirus, or a arenavirus, or a rhabdovirus like a VSV. They are viruses with very different biologies, but all of them, they get out of the cell using a similar concept, is to hijacking this particular pathway of the cell by interacting with different components of the escort complexes. So in the case of the arenaviruses, we and others have identified a variety of interacting partners. So we know that the C protein interacts with TSG101, interacts also with PS4AB, with CHIM3. So as you can see, there are different interacting partners. And because the complexity of the pathway, the question is how many others interacting factors are going to be. The idea behind is that the more interacting partners you identify and you characterize that interaction, more chances to come up with potential therapeutic interventions, basically with compounds that could disrupt the particular interaction. And to address that, we developed an assay amenable to semi-high throughput budding assay. So because the classic budding assay is basically a biochemical assay. You produce the corresponding virus-like particles, and then you analyze those by Western blot, which obviously is not amenable to high throughput. So for that, we explored a variety of possibilities, and finally what we found is that you can generate a fusion protein between the Z protein and Gaussian luciferase in such a way that this fusion protein can be secreted from the cell via budding. Actually, it's not secretion per se, it's the protein gets out of the cell because the C protein, as a bona fide matrix, can be released from the cells associated with membranes without killing the cells. And obviously, as you can see, if you do the biochemical assay, you can see that in the VLPs, you can detect the C Gaussian luciferase fusion protein. You know that this is really operating as it should, because if you use a mutant of the C protein, a glycine in position 2 mutated to alanine, you don't have budding. The reason for that is because the Z protein has to be myristylated in position 2, glycine position 2, to be able to bud. So if you mutate that myristylation site, you prevent budding. So the biochemical assay correlates very well with the reporter gene assay. As you can see, if you have basically the C mutant G2A, you don't get budding. When you have the wild type, you get budding. And if you use a typical inhibitor of myristylation, hydroxymyristic acid, you can prevent the budding. So basically, you have an assay now that is amenable to semi-high throughput assays to identify either compounds or specific host factors involved in the budding process. And one of the things that we found early on is that inhibitors of the PI3K pathway were important for C-mediated budding. So as you can see, a typical inhibitor of the PI3K kinase, LY29402, inhibit very robustly budding by Z. More interestingly, there is a compound called BEC235 that is an inhibitor of both PI3K and mTOR that is currently in clinical trials for treatment of breast cancer, certain type of breast cancer. And this compound also inhibits very strongly budding by Z. This is not due to the effect on mTOR pathway because rapamycin, a typical inhibitor of the mTOR pathway, doesn't do anything to the budding. So based on this, we tested the compound in the context of the natural infection in culture cells, and the compound inhibits very well LCMV. This is a log scale. You have to keep in mind that. You're talking about 3 logs here. It's 1,000-fold decrease. Inhibits also Lassa and Hunin virus. These assays were done in collaboration with our colleagues at UTMB Galveston because they require BSL for laboratory conditions. So, but as you can see, the same compound, as predicted, is hitting all 3 different viruses because it's inhibiting C-mediated budding. So I think this is an illustration how, if you have the right assays, it's now feasible to get into the so-called repurposing of drugs. You can really just go into drugs that are being used for other purposes that they have already cleared many of the FDA regulatory aspects, and then you can try those in your assay and see really what happens. The other component of the virus that you need for production of infectious particles is the glycoprotein. And not only that, the glycoprotein, the GPC precursor, has to be processed into GP1 and GP2. So we know that if you have a mutant glycoprotein that cannot be processed, the glycoprotein reaches the cell surface but cannot get into the particles, and you don't get infectious particles. The processing is mediated, the cleavage of GPC into GP1 and GP2 is done by a cellular protease called the Cy1 protease, S1P protease. And basically, we have tested many of the arenaviruses, and all of them, they use the S1P protease to process the GPC. So the idea is that you may be able to target this step as a general strategy to target arenaviruses. In the case of the S1P, it's interesting because being educated in the ribovirus world, you would say, well, I would bet you good money that if you force selection, if you put the virus in an environment where the S1P is not present, the virus will come up with an alternative to the S1P. Well, every time that we have tried to do that experiment, what happens is that the virus disappears. So we can grow the virus in cells that they do not have the S1P protease, and consistently the virus can replicate very well early on because replication is not affected, but when you do serial passages, this is just one example of many, many replicates. If, when the virus is on the way out, you provide the S1P, then you can restore replication, propagation. So clearly, this is something that still we don't understand, but seems like the virus is incapable to come up with an alternative to the S1P protease, and therefore it's becoming a fantastic target. So actually, there have been in the past several compounds that they have been shown to inhibit the S1P, and therefore inhibit also Lassa multiplication. The problem with those compounds is that they are peptide-based and difficult to really think about how you can really use those in the context of a natural infection in vivo. However, recently, a screen of small molecules by Pfizer identified a compound, PF42942, that inhibits very well the activity of the S1P of the cellular substrates, and consistent with that, also inhibits very well the processing of Lassa GPC or LCMV GPC. However, as predicted, is unable to inhibit the process of a GPC where you have substituted the S1P recognition site by the recognition site of furin. So basically, it's telling you that this compound specifically is targeting the process by the S1P protease. And the compound is capable to inhibit multiplication of LCMV in culture cells. This is in the absence of the drug. When you put the drug in the culture cells, there is a tremendous drop in virus multiplication. If you use a recombinant virus where now you have eliminated this, the recognition site for the S1P protease, and you have introduced a furin site that allows the same thing, allows the GPC to be processed into GP1 and GP2. Now this virus became resistant. What is telling you that the drug is actually acting in a specific way. As you would predict, not only inhibits LCMV, but also inhibits very well Lassa and Junin virus. So because all three viruses, they use the same pathway to generate the GP1, GP2. So basically, as you can see, the more you understand the molecular and cell biology of these viruses, the more chances you have to come up with possible ways to target the multiplication or the life cycle of the virus. And one of the concepts in virology in terms of targeting viruses is combination therapy. So the problem with RNA viruses is that sooner or later, in most cases, it doesn't matter what kind of really compound you use, the virus is going to be able to explore the sequence space to come up with a mutant capable to escape. So the idea is that if you hit them with 2 different drugs or 3 different drugs, and ideally drugs that they target different steps, the chances for the virus to come up with an escape mutant are significantly lower. And this is what you can see here. So this is an experiment where if you treat just with one of the drugs, with the inhibitor of the S1P, as you can see, the virus starts to go down, but very slowly. If you treat with the inhibitor of the replication, the ST545, it starts to go down, but eventually rebounds, and rebounds because there are resistant viruses coming up. Now, the interesting part is if you treat the infected cells with both the ST545 and the PS42, then the virus very quickly disappears. And then if you interrupt the treatment here, you never can rescue virus. So basically, there's a complete extinction of the virus replication. So in the case of Lassa, in particularly, the use of compounds that they can reduce viral load is really very well justified because clinical evidence indicates that the viral load is one of the best predictors of disease outcome. So the clinicians, they will tell you, if the viral load, when the individual walks into the clinic, is very high, the outcome is going to be bad. If the viral load is below a certain level, the outcome probably is going to be a good one. So the idea is that you don't have to have a drug that will entirely inhibit the virus. So perhaps by just dropping viral load by 1-2 logs could be sufficient to then allow the infected individual to mount the appropriate immune response, and the individual will be able to control and eventually eliminate the virus. And as a matter of fact, in the case of Lassa's, some other hemorrhagic arenaviruses, it is clear that during the early days of infection, something happens that the virus can multiply out of control, suggesting that the virus somehow can overcome some of the innate immune defense mechanisms. On the other hand, in the natural environment, what I mean in the natural host, these viruses, they produce a chronic, long-term persistent infection, like for example in the case of LCMV, where you have high levels of replication, widespread high levels, and most of the tissues are involved. This is an example of LCMV in the brain, in the hippocampus area, as you can see, is loaded. And then the question is, so the virus is replicating there, so suggesting that somehow the infected individual or infected cells, they cannot really trigger an appropriate response, especially the interferon response, to control and eliminate the virus, suggesting that arenaviruses somehow they can prevent the induction of type 1 interferon. And to assess that, we use a bioassay. So the concept of the bioassay is that cells such as the long epithelial A549 cells, is a human long epithelial cells, they are very susceptible to induce a very strong interferon response following transfection of DNA and liposomes. It doesn't matter which mixture you do of liposomes and DNA, in such a way that then, upon transfection, these cells, they become highly resistant to a virus that is susceptible to interferon, such as Newcastle disease virus. So as you can see, if you don't transfect the cells and you infect with a Newcastle disease that expresses GFP in such a way that you can monitor infection easier, the infection is very robust. If you transfect the cells and then you go 24 hours later, the virus multiplication is totally inhibited because these cells are now producing interferon, and the interferon establishes the antiviral state. Now, if the cells have been infected with LCMV prior transfection and then you transfect, and then subsequently you go with Newcastle, Newcastle can replicate very well in those cells because LCMV prevents the induction of type 1 interferon. We know that this is due to LCMV because if you cure the cells from LCMV, then the cells, they restore the ability to produce interferon in response to the transfection protocol, and they become again resistant to Newcastle disease virus. So although the virus can prevent the induction of type 1 interferon, it does not prevent the signaling by interferon, because if you treat infected cells with exogenous interferon, they respond, as you can see, by induction of typical ISGs. So basically, these cells that they are infected, they are able to respond to signaling by the interferon receptor STAC-JAK pathway. Cells usually detect RNA viruses in the cytosol using the so-called pathogen receptor recognition sensors, RIG-I and NDA5. These receptors, basically, they can identify unique features in non-self RNAs, such as viral RNAs, and upon recognition of the RNA, they become activated, and then they interact with the mitochondrial signaling adapter protein, MAF, also called IPS1, and then MAF aggregates. Basically, there's an aggregation event and creates a signaling platform by recruiting many different signaling molecules that include the classic IKK pathway involving the IKK alpha and beta kinase and NEMO, and also the non-classic IKK-related kinases involving TBK1 and IKK epsilon. So the canonical or classic IKK pathway is the one that is involving NF-kappa B activation, whereas the non-classic pathway directly activates IRF3 via phosphorylation. So the possibility was that perhaps these sensors, they cannot really see LCMV RNA at all during the infection. That doesn't seem to be the case, because if you do a careful kinetics of the infection, what you see is that very early on during the infection, there is a regulation of type 1 interferon that goes down very quickly once the MP start to accumulate. These levels, they seem to be high, but keep in mind that this is RT-qPCR. However, the levels, they never get to be high enough to have any bioactivity. Basically, there is no antiviral state that is being established with those levels. So because it seemed like the accumulation and the MP correlated with the decrease of type 1 interferon, we tested individual viral genes to see whether one single viral gene was capable to prevent the induction of type 1 interferon. And what happens is that the MP prevents phosphorylation of IRF3, and as a consequence prevents the nuclear translocation of IRF3. So basically, I'm not sure how well this is seen, but when you have cells that they are really expressing the viral nucleoprotein, a IRF3 GFP cannot get into the nucleus. However, in this case, this cell that is not transfected, doesn't have the MP, the IRF3 can get into the nucleus following stimulation with sendai virus. So basically what the nucleoprotein is doing is preventing IRF3 phosphorylation and nuclear translocation. So we have done a comprehensive mapping of the anti-interferon activity within the MP, doing both N and C terminal serial deletions as well as individual amino acid substitutions. And the activity was mapped to the C terminus of the MP. And interestingly, the structure determined by Erica's lab identified within the C terminus of the MP of LASA, in this case, a 3'5' exonuclease domain of the type DEDDH. So this is very interesting because, to my knowledge, only arenaviruses and SARS coronavirus, they have such an activity, a 3'5' exonuclease. So in the case of the arenaviruses, based on sequence, all of them appears, or non-arenaviruses, they have a conserved nuclease, 3'5' exonuclease motif. And also, in the case of LCMV, consistent with the data from Erica's lab with LASA, as you can see, if you mutate each one of the residues located within the active site of the exonuclease, you eliminate the exonuclease activity. So in the case of LCMV, MP behaved like a Lassa MP. And in the case of Lassa, the exonuclease activity was linked to the anti-interferon activity of the Lassa nucleoprotein. And in the case of LCMV-MP, it seems to be also the same situation, because if you have cells that they have a reporter gene, RSP, under the control of interferon, and then you have the cells transfected with a variety of nucleoproteins, either the wild type or nucleoproteins with mutations within the active site of the exonuclease motif, as you can see, in response to Sendai virus, those cells that they express the nucleoprotein, the wild-type nucleoprotein, do not upregulate RSP driven by interferon. However, cells transfected with each of the mutants within the active site, they upregulate RSP. So again, it seems to be that this exonuclease correlates very well with the anti-interferon activity of the MP. So then obviously one could imagine the following situation, that somehow the nucleoprotein prevents the RNA from LCMV or other arenaviruses to be sensed by RIG-I, and because you stop the signal pathway right here, you are not going to get IRF3 phosphorylation, nuclear translocation, and induction of type 1 interferon. And actually, consistent with that, we generated a variety of recombinant viruses with mutations in the active site of the 3', 5' exonuclease. This is just an example. As you can see in this case now, when you have this mutant virus, if you infect the cells with this virus, basically these cells, they sense the RNA, the virus cannot prevent the induction of type 1 interferon, and then if you try to infect them subsequently with VSV, the cells are resistant. And as you can see, there is no cytopathic effect by VSV. However, if you do the same thing with the wild type, if you infect the cells with the wild type, and subsequently you come with VSV, VSV can replicate perfectly well, because the wild type LCMV does not prevents the induction of type 1 interferon. So this correlates very well with all the cell-based assays using the individual nucleoprotein. So we also use these recombinant viruses to examine the role of this anti-interferon activity in the context of the natural infection in the mouse. So we put those into the backbone of one of these immune suppressive viruses. So with the idea to see now if a immune suppressive virus has a mutant glycoprotein that is unable to counteract the induction of type 1 interferon, can it still persist? Well, none of them was able to persist, but the interpretation of the result was complicated by the fact that mutations within the nuclease active sites also have a tremendous effect in viral thickness. And that complicates the whole thing, indicating that we, at that point, hadn't segregated between the anti-interferon activity and thickness. So, and therefore, the interpretation of the results are questionable. So because of that, we just really went ahead and we did a PCR-based quasi-close to saturation mutagenesis of the C-terminus to try to identify mutations that they affected the anti-interferon activity without affecting viral thickness, trying to segregate both functions. And well, we didn't get there, but we found something that really has complicated even more things. We found mutants, such as this one at position 373, isoleucine to valine, that lost completely the exonuclease activity, but retains the anti-interferon activity. So now what we see is that perhaps it's not just the exonuclease activity what is involved in the anti-interferon activity of the MP. So it is possible that the MP also is doing something downstream that prevents the phosphorylation of IRF3 and the nuclear translocation. And therefore what we did is to use coimmune precipitation assays to investigate the interaction of the MP with the different elements of the cascade of RIG-I math. And what we found is that interacts very strongly with IKK epsilon, but not with TBK1 or any other factor of the cascade. So this is co-IP, and as you can see, when you have IKK epsilon and MP, there is co-IP, but MP and TBK1 do not co-IP. So furthermore, we have mapped the interaction to the kinase domain of IKK epsilon, which raises the possibility that perhaps the MP is behaving as a pseudo-substrate for the kinase, for the IKK epsilon, and compete with IRF3. Or alternatively, that the MP inhibits the kinase activity of IKK epsilon. Well, the answer is the second. So basically, if you purify from transfected cells either IKK epsilon alone or the complex MP IKK epsilon, and then you do a in vitro kinase assay using IRF3 as a substrate, what you can see is autophosphorylation of IKK epsilon that has been very well documented. However, this autophosphorylation doesn't happen when you have IKK epsilon complex with MP. You can see how IKK epsilon can phosphorylate IRF3, but no when it's complex with MP. So basically, the interaction between MP and the kinase domain of IKK epsilon is inhibiting the kinase activity of IKK epsilon. And we extended this analysis to other mutants, and what we found that there is a very good correlation between the magnitude of the interaction between MP and IKK epsilon and the anti-interferon activity. Basically, as we indicated before, all the mutants within the active site of the exonuclease, they don't have exonuclease, they do not bind very well at all IKK epsilon, and they do not have anti-interferon activity. But every time that you have a nucleoprotein capable of binding with high affinity to IKK epsilon, you add anti-interferon activity. So it seems like the nucleoprotein of LCMV, based on the data that we have from other arenaviruses, can do more than one thing to prevent the induction of type I interferon. It can probably, the exonuclease plays an important role by diminishing recognition by RIG-I, but then the nucleoprotein also can directly inhibit phosphorylation of IRF3 mediated by IKK epsilon. We haven't had time to get into that, but also the nucleoprotein inhibits the NF-kappa B pathway by preventing the nuclear translocation of the P65 subunit on or NF-kappa B. So the question is, obviously the virus is doing this because it's trying to escape or trying to minimize the antiviral state induced by interferon. Well, the problem with that is that LCMV, as well as other arenaviruses, are highly resistant to type I interferon. As you can see, if you do a dose response with interferon, VSV is inhibited. Again, this is log. You're talking about 4 logs, so you're talking about 10,000-fold decrease production of VSV, whereas LCMV basically doesn't change. So then you say, well, perhaps the virus is somehow inactivating all the ISGs that they are induced in response to the treatment with interferon. That doesn't seem to be the case, because if you have cells that they are expressing a HCV replicon here, and also infected with LCMV, you can do this because LCMV doesn't kill anything. So as you can see, when you treat with interferon, you eliminate very quickly HCV, but you do very little to LCMV. So basically, in the cells that you treat with interferon, there is the ISG responses, the antiviral state is created, eliminate HCV, but doesn't touch LCMV. And the same happens if you have cells infected with LCMV and then subsequently infected with VSV, with pretreatment or not with interferon. If you have cells infected with LCMV and you treat with interferon, you inhibit VSV. So again, basically the LCMV somehow appears to be insensitive to the activity of the ISGs. So then the question would be, why then the virus bothers trying to counteract the induction of type I interferon? It doesn't need it. Well, I think you have to think about what is the biology of the virus. These viruses in nature, they create persistent infections in the natural reservoirs. And if you have a chronic upregulation of type I interferon, most likely an NF-kappa B, you're going to induce probably high levels of apoptosis that they are not compatible with the physiology of the infected organism. And actually, we have some evidence for that, because if you use one of these mutants that doesn't have the exonuclease activity and anti-interferon activity, what happens is that you see a significant increase in apoptosis, an XIM5 IP +2 over time, whereas the wild type, pretty much nothing. So now it starts to really decrease after 10 days, and the reason why this happens is because the mutant virus starts to revert to the wild-type virus because it has an impaired fitness, and obviously over many rounds of replication, so it doesn't have a chance against the wild-type. So another aspect that we are very interested in is trying to optimize ways by which we can really screen for both host factors that they are involved in the life cycle of the virus, as well as compounds that they can inhibit the different steps of the life cycle of the virus. And for that, it would be pretty handy to have recombinant viruses expressing reporter genes that will allow you to do really rapid screens and quantitatively. So for that, we developed the so-called tri-segmented platform for LCMV. So the concept is that arenaviruses are bisegmented, but in principle, you can divide the S segment into two S segments. One of them will produce the glycoprotein and a gene of interest, and the other will produce the nucleoprotein and a different gene of interest. And then what you have to do is you have to deliver both S together with the L into the cells. And then you ask Darwin to be right. So you ask selection to take course. And what happens is if the virus is not capable to pack all three segments, it's going to be missing one gene, and therefore the virus won't go anywhere. So, and indeed this works. The surprise perhaps was that it works extremely well. That basically the virus grows in culture cells as the wild type, and they're very stable, both genetically and any reporter gene that you use, either GFP or in this case chloramphenicol acetyltransferase, over different serial passages, the reporter gene activity is stable. So obviously with this kind of viruses now you can optimize ways to screen for compounds. And just a very brief example, this is something that we did with Casey Nicolau. We screened a library of bijujanagin-related compounds, and as you can see very quickly, you can identify some that they inhibit very strongly the virus, others intermediate, and some of them that they don't do anything. So obviously this is the beauty about having now reporter genes. We have also expanded the use of this tri-segmented platform to try to come up with possible new vaccines against arenaviruses. And in this case, what we did is we moved into the live-attenuated Candid 1 strain of Junin virus. Junin is a highly pathogenic virus. You have strains such as Romero that they are really, they kill guinea pigs within 15, 20 days. But there is a vaccine, a strain called Candid 1 that has been licensed in Argentina, and it seems to be pretty effective. There are some issues with the vaccine, is that genetically it's not well characterized and is heterogeneous. So if you do clonal analysis of the master virus C, you identify clones that they have significant increase in virulence, which eventually is calling for bad news down the road. So what we did is we recreate what we have done with LCMV. We generated a molecular clone of Candid and also a pathogenic virus. In collaboration with our colleagues in Galveston, we verified that the Candid can protect guinea pigs against a challenge with a lethal Junin virus, such as Romero. And then we also demonstrated that you can generate tri-segmented Candid viruses. So in this case, it's GFP. And as you can see, basically, you can generate this virus that they express GFP. So once we felt that we have everything in place, what we did is we generated a Candid that has a mixture of T-cell epitopes identified by bioinformatics by Alexander Settle and Weinbuch-Meyer, so that they cover many different strains of Lassa. So, and as you can see, if you immunize guinea pigs with this recombinant virus, so basically they are fully protected against Lassa and against Junin. Against Junin makes sense because basically this Candid has all the genes from Junin, the attenuated form. But the interesting thing, this is still preliminary data, but basically the initial experiments indicate that it would be feasible to have a platform where you can immunize against Lassa and Junin at the same time. The beauty is that Junin is not present in Africa, and therefore pre-immune problems are impossible, basically. So which obviously offers some advantages over other possible platforms. And basically, we get to the most important slide. So I just want to really acknowledge people that they have been involved in these studies. There are two former members that they play a critical role. Shuso Urata, he was responsible for developing all the assays related to budding of arenaviruses, perfecting the high throughput screen and doing the initial screens. Sebastian Emonet, he developed all the tri-segmented platform for LCMV and subsequently started with the Hunin virus. So we haven't talked about what Masaharu Iwasaki is doing now in the lab, but he's going to be very involved in trying to define the interactome of the L protein, what is interacting within the cells, because something that we didn't have time to talk about it is that between an immune suppressive variant and the wild-type variant, there is a single change in the polymerase that determines that phenotype, that is critical for that phenotype. And obviously, we are interested in trying to understand what differences are in terms of how the virus interact with the host. So Himanja Jajakar, that recently left the lab, unfortunately, she was very instrumental in all the characterization of the MP mutants in terms of the anti-interferon activity, viral thickness, and exonuclease activity. And Beatrice Cubitt, she has been involved in almost in one way or another in all the different aspects for many years now with the help of Nick Knott and Jeff McCuff. And there are many collaborators, so without them, we wouldn't be able to do anything. I definitely have to mention Stefan Kunz. We have been now for many years interacting when he was here before and now that he's in Lausanne. So all the studies related to the glycoprotein, targeting of the glycoprotein are done in collaboration with him. Luis Martinez in Rochester, all the studies related to innate immunity, we are conducting those with him. Dorian, fortunately he left a few years ago, but now we are collaborating using these recombinant viruses. Basically, he uses virus with different colors to look for what happens in real time during meningitis caused by LCMV, because now he can mark T cells and viruses, different things. So I briefly mentioned our collaboration with Kathy Borden in terms of the NMR structure of the Z protein, and currently we are very involved with Sloan Passler at UTMB, because all the experiments involving Hunin, Lassa, any of the BSL-4 arenaviruses, they have to be done there. So great colleagues. I haven't talked about what Michael and we have done in and out all the time, but basically different aspects of the biology of LCMV using now this reverse genetics platform. I briefly mentioned what we did with Casey Nicolau. We haven't had time to talk about some of the studies that we have done using Dale Boger's libraries. With John Jace, we did a proteomics of the C protein that we haven't have time to talk about it, that have identified very interesting partners of the Z protein in infected cells. And finally, with Esteban Domingo, we are still pursuing the concept of using lethal mutagenesis as a element of combination therapy to target riboviruses.